Good afternoon, everyone. I'm going to get started while people are still trickling in. My name's Tandra Fraser, and it's my great pleasure today to introduce Dr. Janet Jansson. She is an Emeritus Chief Scientist and Laboratory Fellow at the Pacific Northwest National Laboratory, where she served from 2014 to 2021. And previously, she was Professor and Vice Dean at the Swedish University of Agricultural College, Sciences, Senior Scientist at Lawrence Berkeley National Laboratory, and Adjunct Professor at UC Berkeley and the University of Copenhagen. Her research focuses on the impact of perturbations such as oil spills and climate change on environmental microbiomes. Dr. Jansson also pioneered use of molecular microbial ecology to study the human microbiome, and she's made considerable advances in understanding the impact of climate change on permafrost and grassland soil microbiomes. Would you like to come up and get started, Dr. Jansson?
Thank you. That was a perfect setup for what I'm going to talk about. I'm extremely thrilled to be here. I've never been in Victoria before. It's just gorgeous. I got to walk around and see some things during my lunch break. This conference is just like being a kid in a candy factory because everybody thinks the same way as I do. It's just great. And I have to point out my background on my slides. It's got to be the cutest and happiest, diverse little soil microbial community that ChatGPT made for me. So I'll be talking about how climate change influences the soil microbiome. So I don't think I need to convince anybody in this room that climate change is happening. It's real. We can see the temperature, global temperature last year. I mean, we keep breaking records for having hotter, warmer global temperatures. The Keeling curve keeps going up. We're having increasingly severe fires. And I know Canada was really slammed with a lot of fires last year, and this year in the winter. In the US, we were getting fires across the western United States. So it's really happening. So there have been a variety of publications and action calls that have been published, and these are some of these thinking about how can microorganisms in the microbial community address some of the challenges that we're having with climate change. So there have been calls to action, scientists warning to humanity. This one with all the citations, let me see my pointer, is that this? Yep. This one was published across all the major journals. Different microbiology societies agreed to publish the exact same article across different journals because they recognize the importance of doing something to address the looming catastrophe that we're facing because of climate change. And I don't mean to say that microorganisms are the only solution, but they're part of the solution, and they should be considered as one of the tools that we can use to help mitigate climate change. The societies, after those publications were released, several societies got together and said, well, we need to actually do something now. And this is a consortium that is currently being developed. It's a community, Microbes Without Borders. We're still thinking about a name for this group, but it involves the major microbiology societies, international microbiology societies that want to do something actionable, to speak with one global voice, to embed microbial science in climate policy, to tell the story. So here's the education, both to universities and to the general public, and to launch high-impact demonstration projects. And so this is a list of some of the societies over to the right that are involved, and I am currently leading the one called Soil Stars. Probably not many of you are familiar with that group yet. It's a new group, but I'll tell you a little bit about it at the end. So the soil microbiome, why do we care about soil microorganisms? Well, one reason, and there's many, but one reason is that the soil microorganisms are really the gate that control the soil carbon cycle and greenhouse gas emissions. And so if we look at this, this nice figure, we can see that plants and some autotrophic microbes take up CO2 from the atmosphere. That is, goes into root exudate. Some of those complex and simple compounds are consumed by the microbial community. You can think of it as the microbial factory that is churning that carbon that originates from plants and other organic material. Some of that carbon is more stable and can be associated with minerals and form part of a stable carbon pool. At least it's a more resilient carbon pool. Part of that is due to the microbes themselves, that when they die, they produce something called necromass, which is a nice way to keep the carbon in the soil. And of course, on the other end, microbes, as they decompose these carbon compounds, and particularly the more simple, easy to reach compounds, they produce carbon dioxide and cycle it back into the atmosphere. And so this microbial factory is really a key, and of course it varies across different soil systems, but we need to understand that how the microbes actually convert and store soil carbon. So this is a talk about climate change. This is a figure that we published a few years ago because we were trying to think how climate change is so many things. And depending on the type of climate change, it influences soil microorganisms differently. So in the center of this little figure, this is supposed to be a mock microbial community. You have fungal hyphae in green and a couple of different kinds of prokaryotic species. And depending on whether you have drought or an increase in temperature or permafrost thaw, those organisms are going to react differently. So for example, with increased temperature here, we often see increased respiration. And so that is a decrease in soil organic carbon. Whereas if you have permafrost thaw, which is here, you can have some organisms become more active. These red moving cells are supposed to represent perhaps methanogens that become activated and produce methane. So we need to think about each climate variable, whereas at the same time this morning I saw some great talks because often they're not in isolation. You can have drought and increased temperature effects that you have to consider together, and how does that impact the microbes. So one thing that we do know is that you will often have diversity shifts, not always, but usually some shifts in the diversity, either evenness or richness, some compositional shifts. Often in hot, dry conditions, the actinobacteria are the super survivors, and they form resistant cell bodies that can withstand those conditions, and so those are often enriched under hot, dry conditions. Whereas in permafrost, as it starts to thaw, you can have an enrichment in methanogens, especially under anaerobic conditions. So another this is a scary ChatGPT diagram. I asked it to show me climate change, and this is what it came up with, but maybe this is what we're looking forward to in the future. What are the consequences of climate change on soil microbes? I think the question should not be, will microbiome changes happen? Because they will. The first question should be, what are the consequences of change? And here I'm introducing a term, it's the metaphenome, which are the combined phenotypes of the community. So really the function, how does the function change? And then the next question is, if it's a bad change, what can we do about it? So I will be talking about two different types of soil systems. The first is permafrost soil, and the second is agricultural and grassland soils. So permafrost soil, that's that was the first pending doom for climate change that got me really interested, because it's really a ticking time bomb. There is so much carbon currently stored in permafrost, as much as in currently in the atmosphere and vegetation combined, and it's like in a deep freeze. And as the permafrost thaws, that carbon becomes accessible to microbial degradation and greenhouse gas production. So I'll be talking about that. I'll also be talking about agricultural and grassland soils. This is my current passion, because these soils, they are a huge reservoir of terrestrial carbon, and they're currently being mismanaged and aggravated by climate change, the soil ecosystems. And so I'll go into that in some detail. Then I will include some information about viruses, and then talk about a few potential mitigation strategies. So I'll start with permafrost. So the permafrost is thawing already in the Arctic. I mean, the Arctic is warming faster than any other region of the globe, and that is resulting in a lot of these interesting landscape features. Coastal erosion is happening. These thermokarst lakes, ice wedges, they're surrounded by ice wedges. You can get these polygon features, kind of look like puzzle pieces. A lot of erosion, and then what we're interested in is what's happening below ground here to the microbes. So we can zoom in on that. This is a cross section of permafrost from the microbial point of view. So on the top of permafrost, you have the active layer. And the active layer is the soil that is seasonally frozen and thawed. So it's not permanently frozen. And underneath that, you have the permafrost. And to be defined as permafrost, it needs to be frozen for at least 2 years. And as permafrost thaws, the active layer gets deeper and deeper, and the microbes that are in the permafrost or going down from the active layer have access to the carbon that was previously frozen. So that means that you have a lot of microbial processes that can occur, a lot of degradation, both aerobic and anaerobic degradation, depending on the soil conditions. For example, these thermokarst bogs are usually anaerobic, and we have a lot of fermentation and methane production. But you can also have CO2 being emitted due to microbial respiration, both aerobic and anaerobic. These are the sampling sites that we've been working on, my group in collaboration with many other teams. So we had several sites in Alaska. I'll primarily be talking about those today. Also Svalbard, here they're sampling in Svalbard, getting some cores. And in Greenland, which I actually got to go to and feel super cool, because I'm on a little boat going by these ginormous icebergs. That was fun. So those are our sampling sites. So this was our first study of permafrost thaw, and we used the metagenomic approach. I'm not going to mention all of my collaborators and postdocs and students that worked on these studies for the sake of time, but I do have them, not always, but I've tried to include photographs in their current positions when possible. But this work was using metagenomics. It was several years ago, but just a simple study. If you take permafrost and thaw it in the laboratory,